Thank you for the introduction. It's my third time here. I'm 3 for 3, and it's always nice to make new friends and meet old friends, which is really what this symposium has been about for me, as well as learning a lot of exciting science. I also want to say that a lot of science is about improvising, and I see we're doing a little improvising here today, so I think that's keeping in the theme of what a lot of scientists have to do on a daily basis. So the title of my talk is Preventing a Disease of Our Own Doing, and the picture that I have on the slide here is from Pittsburgh in the 1940s, at a time when we actually valued making steel more than we did breathing clean air. And I think this speaks to a larger problem in society about how we often can be our own worst enemies, but that doesn't mean there's no reversing that. There's actually always the possibility of making things better. And that's what our lab aims to do. Next, please. So I'll get right into it. So what is the pancreas? The pancreas is an organ which sits in the upper abdomen, and it's actually a gland. Next. And it's a gland because it secretes things, two things in particular, one of which are hormones which help regulate your blood sugar, and those are secreted by what are called the islets of the pancreas. Next, our lab actually is concerned with the other part of the pancreas, which is called the exocrine pancreas. And those are comprised of acinar cells, and these acinar cells secrete digestive enzymes which help you digest your food after a meal. Next. And this is really the focus of our lab. We delve into the acinar cells as opposed to the islets. And the disease that we study is called pancreatitis. And to tell you a little bit about what that disease is, I'll point you to the top left picture, which is what a healthy pancreas would look like. And the top right picture is what a diseased pancreas, or a pancreas that's undergone pancreatitis, might look like. And a big question in the field is, how do we go from the left picture to the right picture? What is the link between a healthy pancreas and a diseased pancreas? What are the mechanisms at a cellular level? Next. So it starts in the acinar cell, the cell I was referring to previously, and there's several injurious agents that can cause disease. Chief among them is alcohol. Previous slide, please. Alcohol is a chief component of injury. Alcohol abuse is one of the leading causes of acute pancreatitis. There's other causes, including the presence of gallstones, which cause blockage of ducts of the pancreas and liver, which can cause problems. Certain medications known to treat cancer and seizure medications are known to cause pancreatitis, and also trauma can cause disease. And what happens in the acinar cell is that we usually begin with an elevation in calcium. Calcium is often thought to be something that's just important for your bones, but you might be surprised to learn that it's also a really important messenger within the cell, a lot of different cells, including the acinar cell. And what happens when calcium gets out of hand and it gets too high in the acinar cell is that it causes, the activation, previous slide. It causes the activation of enzymes which normally digest your food, but instead of digesting your food, they digest the organ itself, and that becomes a big problem, and that's where you get a lot of inflammation from in pancreatitis. Next slide, please. Next, please. So a key aim of the lab is to identify the targets of this calcium, trying to provide the link between elevated calcium levels and the activation of enzymes within the cell, as well as injury, inflammation, and the ultimate disease. And so trying to understand that link is critical to our lab. Next, please. We think we've found one of those links, and the name of the protein, as it were, is calcineurin. And I'll talk a lot more about that in the next slide. Next, calcineurin is a protein, and it has a lot of important functions in the cell, which I won't get too much into. Aside from saying that we believe that calcineurin is a key component in the early stages of disease at a cellular level. Next, please. Next, please. the evidence for this from our lab is that if you give mice inhibitors of this protein, what you find is that those animals are protected against the disease. And what we have here on the top left is a healthy pancreas. In the middle is what pancreatitis looks like. And on the right is a pancreas as viewed under the microscope from a mouse which received a calcineurin inhibitor and then received the disease pancreatitis. And what you notice right off the bat is that the picture on the far right looks a lot more like the first picture, the non-injured pancreas, than the middle picture, which is the diseased pancreas. Those data are quantified below. And so this was a key piece of evidence which clued us into realizing that this protein was of key importance in facilitating this disease. Next, please. The current aims of the lab are to try and understand which source of calcineurin is necessary during pancreatitis. Next. There's many different cells in the body which have this protein. It was actually first found in neurons. It's actually how it got its name. Immune cells have a lot of calcineurin, and it serves an important role in those cells. And the pancreas also has, next please, next. The pancreas also has calcineurin, and it may be important there. The question that we ask is which source is important, which source is necessary. And to do this on a technical level is a little bit complicated to describe, so I'm going to build an analogy, and I'll ask that you use your imagination a little bit. Next, please. Say you have a car, and the question that you're asking is which part of this car is absolutely necessary for driving. Obviously, there's a lot of non-mechanical features of a car that don't really have any relevance to the mechanical aspect of actually just doing what you want the car to do, driving it. So if you wanted to answer that question, what you might do, next please, is to remove components one by one. Next, please. And by doing that, what you're left with is the components that may or may not be important. Next, please. Next. And by doing this, we can then more easily ask the question, next, will the car drive? Now, I know this is a little bit far-fetched from what we do in biology, but it's actually along the same lines. Next, please. So in the mouse, when we tease apart the mouse's DNA, if you will, or disassemble the mouse's DNA, just like we did to the car, we ask a very similar question: which source of calcineurin is important? Neuronal calcineurin, pancreatic calcineurin, immune cell calcineurin. And we perform a very similar experiment. Next, please. Where we remove pancreatic calcineurin and pancreatic calcineurin only. And by doing that, we can eliminate one key component of this protein, or one key source of this protein. Next, please. Next. And so we've actually begun these experiments, and what we found is that if you delete calcineurin from the pancreas, and only the pancreas, you see a pretty interesting finding, which is that these mice are protected just as they were when we gave the inhibitor in the previous data set. Even more interesting, next please, if you delete calcineurin from the acinar cells specifically, you also get a reduction. And these data suggest that pancreatic calcineurin is important, and acinar cell calcineurin is also critical. And these studies are ongoing, and we're trying to understand all different sources of this protein and which ones are really critical to the disease. Next, please. Now, this is really the most important slide given the symposium. I'm sorry, I know my time's up, but I just have to talk about this. It looks like I'm up against a restart here as well. I will say that this surah was not... It's well known, and it's from Surah Abasa, and the verse reads, "Does he not consider from what thing did God create him? From a sperm drop. He creates him and proportions him. Then he makes the way easy for him." And that's from Surah Abi. And that word proportioned I wanted to just focus on because it's the one that struck me. The word actually is qadarahu in Arabic, and it's derived from the verb qadara, which has many different translations. Among them is arranging, setting out, planning, programming. And that word programming to me stuck because if something is programmed, it can be reprogrammed, which is precisely what we're doing in these experiments to better understand disease. We're trying to reprogram the genetic material of animals in order to understand which components are absolutely essential. I wish I had more time to talk about this because many thoughts sort of floated through my mind as I was pondering this verse, but I'll let you sort of make interpretations on your own, and again, I think there's many. So without further ado, next please. These are the members of the lab who have done all the work, and as well as Dr. Hussain, who's been the lead. And I also have to thank Dr. Hussain for his mentorship and for inviting me to be here today, and I'll leave you with a picture of Pittsburgh, which is much nicer than the one I started with. So I think the future is bright. Thank you very much.